Hello guys and welcome back. In last week's video, I talked about what is pharmacogenomics part 1. In today's video, I am going to talk about pharmacogenomics part 2. I would like briefly to talk about the pharmacogenomics, and for those who may not seen part 1. Pharmacogenomics is an important example of precision medicine, which aims to tailor medical treatment to each individual or group of individuals. Pharmacogenomics studies how your DNA influences how you respond to drugs. In some cases, your DNA can influence whether you have a bad reaction to a drug or whether it helps or does not help you. Pharmacogenomics can help you improve your health by predicting whether a drug will benefit you and be safe for you to take. Knowing this information can help your doctor find the best medicine for you. How does pharmacogenomics work? Depending on how you take the drug and where it acts in your body, drugs can interact with your body in a variety of ways. Your body needs to process a drug after you take it in order to deliver it to the desired location. Your DNA can influence several stages of this process, and which will impact how you react to the drug. I have some examples of these interactions for you guys. Receptors for drugs. Some medications function properly only when they can bind to receptors, which are proteins found on the surface of cells. Your DNA determines the types and numbers of receptors you have, which can impact how you react to a drug. You might require a different drug, or a higher or lower dosage than the majority of people. As an example, consider breast cancer and TDM1. Some breast cancers produce an excessive amount of HER2, a receptor, which aids in the development and spread of the cancer. TDM1, a drug that attaches to HER2 on cancerous cells and kills them, can be used to treat this type of breast cancer. If you have breast cancer, your doctor may examine a sample of your tumor to see if TDM1 is an appropriate treatment for you. TDM1 may be prescribed if your tumor contains a high amount of HER2, HER2 positive. TDM1 will not work if your tumor lacks sufficient HER2, HER2 negative. Drug uptake. Some medications must be actively absorbed into the tissues and cells on which they act. Certain drugs can be affected by your DNA. Reduced uptake may indicate that the drug does not work as well and may cause it to accumulate in other parts of your body, causing problems. Your DNA can also influence how quickly certain drugs are eliminated from the cells in which they act. Drugs may not have time to act if they are removed from the cell too quickly. As an example, consider statins and muscle problems. Statins are medications that operate in the liver to help lower cholesterol. Statins must first enter the liver in order to function properly. A protein produced by the SLCO1B1 gene transports statins into the liver. Some people have a specific change in this gene that causes less of a statin called simvastatin to be absorbed by the liver. Simvastatin can accumulate in the blood when taken in high doses, causing muscle problems such as weakness and pain. Before prescribing simvastatin, your doctor may also recommend genetic testing for the SLCO1B1 gene to determine whether simvastatin is the best statin for you or what dose would work best. Drug breakdown. The rate at which your body breaks down a drug can be affected by your DNA. If you break down the drug faster than most people, your body eliminates it faster, and you may require more of the drug, or a different drug. You may require less of the drug if your body breaks it down more slowly. Depression and amitriptyline are two examples. The antidepressant drug amitriptyline's breakdown is influenced by two genes known as CYP2D6 and CYP2C19. If your doctor prescribes amitriptyline, he or she may suggest genetic testing for the CYP2D6 and CYP2C19 genes to help determine the appropriate dose. If you break down amitriptyline too quickly, you will need a higher dose, or a different medication to work. If you have a slow breakdown of amitriptyline, you will need to take a smaller dose, or a different drug to avoid a bad reaction. Targeted drug development. Pharmacogenomic approaches to drug development go beyond simply treating symptoms to address the root cause of the issue. Specific alterations, mutations, in a gene are the root cause of some diseases. Different types of mutations that occur in the same gene can have various outcomes. Some mutations could cause a protein to function incorrectly, while others could prevent the protein from being produced at all. Drugs can be developed based on how the mutation affects the protein, but they will only be effective for a particular kind of mutation. Cystic fibrosis and ivacaftor are two examples. Mutations in the CFTR gene, which affect the CFTR protein, cause cystic fibrosis. The CFTR protein forms a channel that serves as a passageway for particles to move across your body cells. The protein is correctly made in the majority of people, and the channel can open and close. Some cystic fibrosis mutations cause a closed channel. The drug ivacaftor works by forcing the channel open in this type of mutation. Ivacaftor is unlikely to help people with cystic fibrosis, where mutations prevent the channel from being formed at all. Any queries you may have should be asked without hesitation. I genuinely hope you liked viewing, and if you are, please giving it a thumbs up, subscribe it, share it with your friends and ring the bell when new videos are become available. I appreciate you for your time and thank you for watching.